I'm going to go back to basics really and just give you a little bit of insight into what we do at Jealott Hill in terms of bringing new products onto the market. This is me. I've been here since before the days that Sarah alluded to in Zenica. I was here in ICI days back in 1985. Started off my career on site, basically been sent overseas on a one-way ticket to do field trials in places like Africa, South America, etc. So I've been around a while, primarily working in insect control. So just to give a bit of background onto where we are here in relation to, and what it does in relation to the company. We're at Jealott Hill, and research into new products takes place basically at Jealott Hill and at Stein. At Jealott Hill, we do the discovery of new active ingredients and herbicides, and then at Stein, we do insecticides and fungicides. So the kind of things in terms of chemistry that we're looking for: herbicides, fungicides, insecticides, and nematicides. We also do work on things we call abiotic stress. So that is things like how to apply something to your crop which makes it more tolerant to water loss or heat stress, etc. And also other things that can affect yield and quality as well. So there is a market for those things just evolving, and we do the research into that as well. So in terms of developing a new product, it's all very, very simple really. All you have to do is answer those 5 questions. Does it work? Is it ours? Can we make it? Is it safe? And then ultimately, we're a commercial company, will it sell? So that answering those 5 questions takes us quite a long time. A minimum 10 years, probably 15 years, sometimes more than that. So where do we get our new ideas from? Now, active ingredients, the inspiration for them comes from multiple sources. Natural products is a big one. We have like the pyrethroid insecticides coming from chrysanthemum flower extracts, mesotrione coming from the bottle brush plant, strobilurin fungicides, again coming from a natural source as well. So natural products is a very, very big source of inspiration for us. Literature, reading the scientific literature, see what's in there, see what other people are working on. Can we do something similar or better? Third-party offers. We do get things from other companies or universities, whatever, saying, this is a good idea, have a look at this. We might take it as it is, or we might work on it further to develop it to something else. Mechanism-based design. So we look at the chemical structure, see how it interacts with the target site in the organism. Can we change it a bit, make it better, quicker, faster, more efficacious? What we call cross-indication screening. Now, that's an interesting one. Many of our insecticides actually come from things which started off as herbicides and vice versa. And if you look through the literature and how products are developed, many of them come from a source which is not the intended area. So a fungicide will ultimately end up as an insecticide. And so that happens in the pharmaceutical industry as well. You see the same thing happening. They thought they were going to invent something as a painkiller, and it turns out to be an anti-arthritic drug or whatever. So that happens a lot. The rest of it, random. We just test compounds that we buy. Some of them are biologically active and interesting to us, and there's a number of our products on the market that have come from that source. Other ones, we do have the geeky scientists here who sit in rooms on their own and think about things. So they will come up with ideas and for new products as well. So that happens. There's a whole range of idea sources. And one of the compounds, the products we're talking about today, chlorantraniliprole, acelepryn, that came from a natural source. So for many years, people were interested in ryanodine. Very nice compound, biologically active, but you can't get enough of it out of the plant to make a product, and also it was toxic as well. So it took a lot of companies a lot of time to actually come up with chemistry that you could make, that was safe, and that worked from that source. But the inspiration there was from a natural source. So this is an interesting one. Is it ours? And this is what, as a commercial company, we are very interested in. If you invent something, you can file a patent, and everything we see around us will be patented. You then have 20 years protection that nobody else can come along and produce the same thing. And so that 20 years, now there's tactics there. Do you file your patent really early, as soon as you discover it? Or do you wait a bit and increase the time that it's protected for? So it's a real balance, because if you wait, somebody else might come along with the same thing and beat you to it, and then you have no control over that, or no comeback from it. So this is an area we concentrate on a lot. We want to protect our products. We also then, after the invention, the actual molecule, we will look at what we call use patents as well. So could you use it for this purpose? And no one's ever thought of that before, therefore we could file a patent for that. Can we make mixtures? That's another thing. If you combine it with something else and it has a surprising effect, you can patent that. So we do a lot of work in trying to protect our products from our competitors doing the same thing. And our competitors do exactly the same. So it's a constant, not a battle, but it's a very challenging environment, and quite an exciting one as well. So we spend a lot of time on this. Does it work? Well, the site that you see around you is basically doing that, seeing if those molecules have the effect that we actually want to get from them. And so the kind of questions that we'd ask ourselves is, of a chemical type, which one of those is the most active? And that would maybe take us 10 years to work out. We'll produce thousands of compounds, all closely related to each other, and we want to pick the most efficacious one, the one that delivers the effect that we want to sell. And then we'd ask, does it have the right biological spectrum? Does it do the things that we want it to do? And then does it work in the lab? Does it work in the field? And sometimes we find that things that work in the lab don't work in the field for whatever reason. They might be not stable in ultraviolet light, outdoor conditions. They might bind to the soil, bind to plants in the wrong way. And so they're not doing the things that we want it to do. Does it work on resistant strains? That's one that we always have to answer. And everything we do in fungicides, herbicides, insecticides, we have to make sure that our new molecules work against those strains that are resistant to chemicals that are on the market already. So that takes a lot of work to do as well. And it has happened occasionally that we spent 10 years working on something. By the time we get it to market, there's a resistance to it already, even though it hasn't been on the market. And so that occasionally happens. And then you've wasted many millions of dollars and have no product for it. We have to know about the mode of action and biokinetics. The biokinetics means how does it move within the soil, within the insect? How does it get across the cuticle of the insect to the target site within the insect? How does it do that? And through our chemistry, we can actually affect that, or through our formulation, we can affect that. So we have to understand that. Where does it go? What does it do when it gets there, and can we influence that? And so we'll have a whole building down the bottom end of the site that just does that for fungicides, herbicides, and insecticides. Lastly, formulation development. What you see in that bottle has surprisingly taken many years to work on. Can we get it to that target site within your target organism quickly, effectively, and without losing anything? And so that's what we spend a lot of time doing as well. We have a whole department over there doing that job. So what you see in the glass houses around you on site, if you have a look out the window at lunchtime, basically is doing this. This is some of our herbicide screens on site. So as we move through the screens, we get to more realism. So these, at the very first stage, we're using small plants in a 96 well plate, just seeing, does our compound work or not? And that's it, purely a simple question, yes or no. If it doesn't work, it goes out. And then gradually, if it works, we'll introduce more realism, more species, and a slightly more realistic scenario. And then if those work, they will go into the glasshouse screens, which is much more realistic. Then obviously, ultimately, you'll go into the field or mini plots or whatever, but you introduce more realism. The reason is the cost of doing this is we are screening hundreds of thousands of compounds a year, and we don't want to invest too much time and effort into that process. We want to make it slick, quick, and cheap. And so you gradually introduce more realism. With more realism comes more cost. And so, as I mentioned, really, this is just a little bit superfluous. Our initial discovery screens, which we do at Jealott's Hill, use a very small, less than a milligram of material to answer that question, does it work or does it not work? And we answer that against fungi, weeds, and insects in the first instance. So one compound will be tested against all of those representative insects, weeds, and fungal pathogens. And very miniaturized, very quick, and enables us to get through a lot of compounds very quickly. Very important is mode of action. Now, the reason this is important, A, we need to understand what our compound does and how it works. Is it a new effect? Is it targeting something new that hasn't happened before? But B, there is a product safety reason, we have to understand, is that way it's working propose any hazard to both humans or other non-target organisms? And so this screen that we do, which tests against multiple different modes of action, different target sites, which occur both within insects, plants and fungi, and indeed people. We tested against this very large battery of targets to determine how it works. Sometimes we get no hits on this, so it's working in another way, but that still tells us something. We have to elucidate how our compound is working. And we see this is a good example of it, ranitidine receptor, calcium channel disruption, and Sean will talk a bit about this in more detail. So right at the early stage when acelepryn was invented, you have to know how it works, you have to know exactly which target site it's interacting with, and one of the challenges is getting it there. So you will adapt your chemistry, alter that molecule to make sure it gets to that target site, and then is the most efficacious it can be when it gets there as well. And understanding that at an early stage in your project helps you to optimize that chemistry and make sure it's doing what you want it to do most effectively. Is it safe? Well, this is probably what adds the biggest cost to the development of a product. This would cost us tens of millions of dollars to do. We have to know if there's any hazard attached to it. Is there a risk to people, to non-target vertebrates, invertebrates, etc.? What does it do? If it's a crop plant that's being sprayed onto a crop, what does it do when it gets on there? Does it break down? Does it disappear? Are there residues there that you could eat? So we have to know that. And so we'll spend a lot of time, a lot of resources doing that, radio label studies, looking at the movement of it within that plant. Does it get into the parts of the plant that you harvest? Is it still there after harvest? If you're making food from it, is it there after you've processed that food? So we'll answer those questions. Ultimately then, are we exposed to it in our diets? And importantly, are workers exposed to it? And if they are exposed to it, is there a risk to it? And so we have to answer those questions. And then where does it go in the environment? If you spray it on your turf, does it go down to the soil? Does it move in that soil? If it does move in the soil, does it get into the watercourses? We have to have answers to those in order to get registration. So we have to basically understand where it goes to and what happens to it on that journey. And we have to account for that, and we won't get a registration unless we can do that in a manner that is convincing to the regulators. What effect does it have on non-target organisms? A very important one. What does it do to the earthworms, the non-target, the predatory mites, the predatory insects, birds? We have to be able to trace the compound through that route. I do some work on things like rodenticides, and you have to show that if a predatory bird eats your dead rodent, the compound in it has no effect on it. And so there's multiple effects you have to account for, and all of those have to appear in your regulatory dossier, which will be mentioned later on in the presentations. Lastly, where does it go? We have to understand all the physical chemical properties of our molecule. What is it like? In terms of, does it bind to the soil? Does it move into the soil? And that's one of the things with acelepryn. We know that it stays in this top part of the soil, and that's down to its physical chemical properties. It can bind to the soil, but it can still be biologically active, and it won't move down into the watercourses. And so we again, very early on in the projects, we'll look at the physical chemical properties. Are they desirable for us? And if they're not, can we change it without altering the biological properties of the molecule? Does it bind to organic matter? That's an important one for us. As I mentioned before, it influences the movement of that compound within the soil. Is it rapidly degraded? Well, for many of our uses, we want it not to be rapidly degraded, but our regulators want it to be rapidly degraded so it disappears quickly. So it's finding that balance there that we have to do. Making sure that it lasts for as long as we want for the biological effect to be exerted, but not too long that it's there the next season as well. Lastly, or not lastly, second but to last, can we make it? There's no point in doing all that work if you can't make it at a price that you can sell. So right from the early stages in a project, we will start estimating the cost of it to produce it. Then gradually through those 10 years or so, we will be making it on a larger scale and getting a better understanding of the cost, and can we produce a factory that would make that? And so that's critical to us. Lastly now, can we sell it? Will it sell? There's no point in doing all that work and then finding that you've got something that no one really wants. And that has happened. Things have disappeared, not so much in recent years, but certainly I remember in the past, things being launched and then taken off the market 2 years later, because no one's really bothered with it. And so you have to have an effect that is desirable to your customers. They have to want it and want to buy it. And that can be down to the marketing or the unique effect that it exerts on the target organisms. Is it technically robust? Does it have a good reputation? And so we have to be able to ensure that our customers understand what it can do and they want to buy it because it does the right thing at the right time and gives the effect that is wanted and ultimately delivers something that is of value to them. Is it cost effective? It's no use being able to deliver those things if it costs 10 times more than they're willing to pay. And then how to look after it in the marketplace. As the product has been on the market for a number of years, it might become less attractive because people are so familiar with it. Can we do anything to revive that? Can we add something to it? Can we make a new formulation, make it more current? And so we'll be thinking about those things after the launch. And so ultimately for a commercial company like Syngenta, that is very, very important. And we have the whole departments again working on that kind of thing and thinking about what do people want, what they want to buy, what will they pay for it, and does it deliver the effect that they want? And so they will continue to buy it. And I think that is my last slide, and I will be handing over to Sean, I think.
Thank you.